name is Rosemary Whittle. I am a PhD candidate at the University of Guelph, and today I'm going to present a case study. Maternal flaxseed diet had sex-specific effects on the weight of broiler chickens diagnosed with novel avian reovirus and infectious bronchitis. I will start by giving a quick introduction to omega-3s, how they influence immune function, before outlining avian reovirus and infectious bronchitis. I will then present my experimental design, results, and conclusions. What are omega-3s? Omega-3s are long-chain polyunsaturated fatty acids. In this presentation, I will focus on just two of these, alpha-linolenic acid, or ALA, and docosahexaenoic acid, or DHA. Omega-3s must be ingested because they cannot be synthesized internally. Birds, including chickens, are fairly unique in that after they consume ALA, their bodies elongate and convert it to a more functional longer-chain fatty acid such as DHA. Therefore, they do not have to directly consume DHA to reap the benefit of it. Sources high in ALA include flax seeds, canola, and chia. DHA predominantly comes from marine sources such as oily fish and algae, which are not regularly fed to chickens due to the effect on the taste and smell of the eggs and meat. There is already a large volume of research that supports the hypothesis that dietary fat consumption has implications on the immune system of mammals and birds alike. This includes direct effects on the cell function, including membrane fluidity, impacting receptors, enzymes, signal pathways, and icosanoid and cytokine production. Icosanoids inhibit inflammation and other immune responses, as cytokines and glycoproteins, which help synchronize immune system functions. Some are pro and others are anti-inflammatory. Omega-3s may also directly affect gene expression related to immune cell function. These effects culminate in omega-3s having anti-inflammatory but also immunosuppressive properties. I'm now going to give a brief overview of avian reovirus and infectious bronchitis. Avian reovirus can be present in the absence of disease. This means that chickens may not be symptomatic but may still carry the virus. It can be transmitted vertically from parent to offspring and horizontally through inhalation and ingestion. It has been known to induce many clinical diseases including viral arthritis, runting and stunting syndrome, malabsorption syndrome, chronic respiratory disease, immunosuppression, and inclusion body hepatitis. The main symptoms of avian reovirus includes poor and deformed feathers, runting, stunting, and poor feed conversion, leading to low body weights. Infectious bronchitis is a form of avian coronavirus. This is a highly contagious upper respiratory tract infection which has a short incubation period. Upon exposure, symptoms begin to present within 24 to 48 hours. Infectious bronchitis is transmitted horizontally through ingestion and inhalation. Symptoms include coughing and sneezing, facial swelling, and reduced weight gain. Here is a quick overview of my experimental design. I raised a flock of Ross 708 broiler breeders in 8 pens. Trial diets were only fed to females. Males were fed a controlled diet throughout. During the rearing phase, from hatch to 16 weeks of age, 4 pens of females were fed a corn, soybean-based control diet. The other pens were fed a flax diet. This was the control diet, but including 2.57% full-fat flaxseed. At 16 weeks of age, 2 pens from the flax and 2 from the control diet were switched to the opposing diet for the laying period. This created a 2x2 factorial design. Eggs were collected at 30 and 33 weeks of age for incubation, and a total of 260 offspring were hatched per maternal diet combination over 2 replicates. All birds were weighed weekly to monitor their growth rate. It is important to note that this is a case study and not a controlled immunological trial. During the rearing of the offspring, there was an outbreak of infectious bronchitis. Symptoms were observed from 1.5 to 4 weeks of age in both replicates. A post-mortem analysis was conducted and it was confirmed that this was the Delmarva variant of infectious bronchitis, which was prevalent in our region at the time. From the onset of the trial, low body weights were recorded from 1 week of age. There was a high percentage of runs and feather deformities were observed, leading to a diagnosis of novel avian reovirus. Post-mortem analysis also found intestinal lesions which are cohesive with malabsorption syndrome. This is a quick rundown of my statistical analysis. The data were normally distributed and a linear mixed effects model with repeated measure ANOVA was used in R, maternal rearing diet or MRD, maternal laying diet or MLD, sex and week were used as fixed effects. Here are my results. The graph on the left depicts the comparison between the average weight per week that I recorded in my trial, shown in blue, and the Ross 708 target weights in orange. We have week of age on the x-axis and weight in grams in the y. As you can see, my recorded weights were almost half of what was expected in accordance with the guidelines. The graph on the right shows the mean weights for each maternal diet combination. There was very little difference between them, however, by week 6, the rearing by laying diet combination of flaxseed by flaxseed offspring appeared to weigh less than the other 3 combinations. But there was no interaction between week of age, maternal rearing diet, maternal laying diet and sex, or even between maternal rearing diet and maternal laying diet. There was a significant effect of flaxseed, maternal rearing diet and sex. The graph on the right shows the sex, F for female and M for male, for each week of age 1 to 6. The weight is on the y-axis and is in grams. A post-hoc Tukey test contrast showed that females tended to be heavier than males on week 1 and were significantly heavier than males on weeks 2, 3 and 4. There was no significant difference between males and females on weeks 5 and 6. I also found no interaction between maternal laying diet and sex. Coefficient of variation was used to analyze body weight uniformity. There tended to be a 4-way interaction between the factors and a 3-way interaction between maternal laying diet, sex and week. However, there were no significant contrasts within week of age in the post-hoc analysis. There was a significant effect of week on coefficient of variation. The graph on the left shows the week of age on the x-axis and the coefficient of variation on the y. The coefficient of variation differed significantly on week 1, 2 and 3 from each other and all other weeks. Weeks 4, 5 and 6 were not significantly different. To better visualize the variation in body weights, the graph on the right shows a frequency density plot of weights on the x-axis and density on the y. Each peak and color depicts a different week of age. Usually, we would expect to see a nice bell curve for each week, but as you can see, the peaks going from left to right of the graph go from mountains on week 1 to rolling hills to flatlands by week 6, as the distribution of weights becomes so dispersed. In order to try and understand my results a bit better, here are the egg fatty acid profiles from my broiler breeder hen eggs. The left graph shows the percentage of total fatty acids for DHA, or docosahexaenoic acid, and the right for ALA, or alpha-linolenic acid, for each maternal rearing by laying diet combination. Looking quickly at these graphs, we can see that flaxseed in the maternal laying diet, depicted in light and dark green, has a much larger effect on the fatty acid profile of eggs than when flaxseed is provided only in the maternal rearing diet, as shown in light blue, and especially compared to the control by control group, depicted in dark blue. In conclusion, feeding omega-3 to broiler breeders during rearing and during lay did not improve offspring performance when challenged with a natural outbreak of disease. Feeding broiler breeder hens the flax diet during rearing reduced male offspring growth compared to females. Does this mean that flax maternal rearing diet had an immunosuppressive effect on males? Reflecting upon my results and the egg fatty acid profile, I came across further questions. Why is there only a maternal rearing diet effect? We already know that life experience of mothers during development, including stressful events, can have effects on the behavior and physiology of their offspring. Perhaps this is also true for maternal nutrition during development. For example, brood size manipulation studies relating to nutritional stress of wild birds such as zebra finches influenced offspring phenotype. This leads on to sex-specific effects. Why did we find a sex-specific effect in this study? Early maternal nutrition, or nutritional stress, of zebra finches also had sex-specific effects on the phenotype of their offspring, producing heavier males but not females under nutritionally adequate conditions. This may be one hypothesis as to why flax maternal rearing diet had an effect on the weight of the offspring. I'd like to give a quick thank you to the funding bodies that support my research. My supervisor, Dr. Tina Wadowski, Principal Investigator Dr. Elijah Chiari, the farm staff at Canadian Poultry Research Station, and the handful of work-study students that were invaluable throughout this trial. Thank you for listening.
All right. Thank you very much, Rosemary. Nice presentation.
Thank you.
So one of the questions that's come up is, are there other factors or other compounds or other components of flax that might also influence the effect of the viral challenges?
To be quite honest, I'm not really sure because I've only looked at omega-3s. Very possibly there's a lot of dietary effects which then can impact immune system, but that's not really what I'm studying. It's just focusing on omega-3s.
All right. Yeah, fair enough. Okay, not seeing any questions right now. So in terms of, and you may have shown this, but in terms of the omega-3 fatty acid composition of the flax diets versus the control diets, how different were they in presumably just linolenic acid?
They were pretty much different, especially between when it was fed in the maternal rearing diet versus the laying diet.
Mm-hmm.
There wasn't really a difference between those who were fed just in the laying diet compared to those which had linolenic or flaxseed from the beginning. So when they were fed it in the rearing, that was similar, and then when they were fed it just in the laying, it was also similar.
Okay, great. So another question coming through. So the results you presented were sort of a case study of what happens when the birds had this challenge that we weren't necessarily planning for. So if you were going back and could do it again without knowing that you would not have this challenge, what would be your intended results, or what was your goal of the study or your hypotheses without this challenge part of it?
So we were mainly looking at sort of leg strength parameters. So there's a lot of research which looks at anti-inflammatory effects of omega-3s. So we weren't looking at weight per se. We were more looking at the additional measures such as gait scoring. We also did water bath tests to test how long they can stand for. Of course, that was all affected by the fact that we had this reovirus. Sorry, my cat. By the reovirus. So the data from that isn't quite so relevant anymore. But we expected the omega-3s to have anti-inflammatory effects and therefore increase leg strength, so have lower gait scores, be able to stand for longer in water, that sort of thing.
Okay, great. So will you have the opportunity to rerun this trial? I know breeder trials are costly and time consuming. So will you be able to redo the trial and hopefully achieve some of those original goals?
Unfortunately, not within the scope of my PhD. I'm also running a trial with laying hens, so that sort of started immediately after the broiler trial ended. So not within the scope of my PhD, but maybe in the future.
Okay. All right. Well, thank you very much for your presentation.